Good morning, everybody. It is my great pleasure to welcome Professor Molly Scheel. Molly is the Navari Family Professor in the Department of Medical and Molecular Genetics at the IU School of Medicine in South Bend. So she's a close neighbor. And I'm delighted that she's able to join us today to share with us really a research journey that she has had from bench to field. Molly describes herself as a geneticist and a developmental biologist, and has been developing some very exciting technology around biorational RNAi-based vector control tools, and really has been working with diverse groups of people, from geneticists to entomologists to people in the field, also looking at engineering and ethics and social science, and technology development and transfer. Just a little bit of background. I've known Molly for a number of years. We keep bumping into each other at meetings and talking about her work. I've read her papers, but have never actually had the chance to hear her present on her work. So thank you so much for joining us, Molly. It's a treat. And just a bit of background. So Molly has an interesting career path here. She started out at the University of Chicago, where you did your postdoc, and then as an assistant professor at Albion, and then at University of Notre Dame, before joining Indiana University, and recently promoted to full professor. So congratulations. The title of Molly's talk is Biorational RNAi-based disease vector mosquito control. I should say also, Molly is very active in the American Society for Tropical Medicine and Hygiene, and currently part of the American Committee for Medical Entomology, plus numerous other accomplishments and awards. But I don't want to steal any more of her time. I'll monitor the chat for questions. If you look in the chat, you'll also see that Molly has provided a link to a video that shows some of her work. And she's also given her email address in case anybody would like to get in contact with her. We're coordinating her schedule, and we do have a group discussion session from 2 to 2:30. If you'd like to join that, please just email me and I'll send you the Zoom link. But I don't want to take any more of Molly's time. I know she's got some really exciting work to talk about. And so I'm going to hand the virtual floor to her and welcome Molly to the Department of Entomology at Purdue.
Thanks very much. Thanks to Dr. Hill for the kind introduction and for the invitation. And I wish we were in person, but I'm glad to be with you in any format. So again, don't hesitate to contact me after this if we don't get to all of your questions, but I will push on with no further ado. Just to give you a little more background, I'm actually a basic scientist by heart, at heart, and although we're gone on this translational journey, in many ways I am not doing too many different things than I did as a graduate student at University of Chicago, when I studied all these different arthropods that are shown up here and more. And I think that comparative developmental genetics flair hopefully will be evident in my presentations today, as we take this on a more of a translational route, which has been a journey for me. But we have been studying primarily, our work is initially in Aedes aegypti mosquitoes. And as you no doubt know from the Zika epidemic a few years back, these are the vectors for Zika virus, chikungunya, yellow fever. as well as dengue, which is probably responsible for most of the deaths by the pathogens vectored by this mosquito. And the take-home message is always, if we can control this mosquito, we can actually get rid of all of the transmission of these viruses as well as future threats. And so targeting the mosquito is really the primary way that we have at this point in time of controlling these diseases. And my background is in developmental biology. I'd done work on many different arthropods. And when I got to study up at the med school, and we're located on the campus of Notre Dame, I knew that I was going to interact with many of the mosquito biologists at Notre Dame. And my goal was really to just characterize mosquito development. And I was shocked when we started that journey to learn that we knew very little about these early developmental and larval pupal stages of mosquito life cycle, primarily because the time when they vector pathogens is, of course, when they're adults. So these were really quite understudied, and we wanted to set out initially to remedy that. I started working with Dave Severson at Notre Dame. He had just done the Aedes aegypti genome project, and it was really, he reached out to me first, and that's historically why we started working on Aedes aegypti. We stayed working on them because they're one of the best mosquito developmental models. They're very easy to rear. They will lay their eggs on a paper towel. Their eggs go through diapods, so you can store them. So if you're doing genetics, you don't have to be like in your fruit fly labs where you're changing over the food every few weeks. You can actually toss them in a drawer and hatch them when you need them. So there's many great things about that. Being a comparative person, then we can take what we learn in Aedes aegypti and translate it to other mosquitoes such as Anopheles, which are much harder to rear, and Culex. Culex are the vectors for lymphatic filariasis, and around here, as you know, for West Nile virus too. And Anopheles, of course, are malaria vector mosquitoes. So as you know, in this department, mosquitoes lay their eggs in aquatic habitats, and the larval stages and the pupal stages are spent in that aquatic environment. If we look at Aedes aegypti, these tend to be found near people's homes and domestic breeding sites, water, in bird baths, or in plant containers, cemeteries, things like that. You'll find water, tires that carries Aedes mosquitoes. And you can easily dump out that water and control some of the mosquitoes that won't be found in tires by getting rid of tires. But when you get to island environments such as Trinidad, where we have a field site, people are storing waters for the dry season in these large containers, and they use it for dishwashing, sometimes for drinking water too. So you need to store that water, and there are, of course, mosquitoes breeding in those containers. So it gets harder to control those mosquitoes if you need that water for living. Problem being, too, that if we look to mosquito control, as you know, pesticide resistance is a key problem, and Aedes aegypti are becoming increasingly resistant to insecticides. This was actually at the start of the Zika outbreak, a map of pesticide resistance in Puerto Rico, and you probably can't see all the details, but all these red areas are places where resistant mosquitoes had popped up and been identified for some of these commonly used pesticides. So it's a real problem, not just in Puerto Rico, but across the globe. And also a big concern, and these are some pictures of when the Zika had hit in Florida, people were really, of course, that they were fuming with these huge amounts of pesticides. People were worried about the impacts on their own health. And so there's a lot of pushback against pesticide use. All of these things have come about while we were going on this journey studying genes in Aedes aegypti. And initially I thought, well, okay, if we just characterize the development, some new ways of controlling mosquitoes might develop. And to some extent that's true, but we really had to sort of think about it in a little bit different way, and I'll talk a little about how that emerged. But initially we were just using RNA interference, or RNAi, for selective gene targeting. And as you probably know, we have a pathway that produces small interfering RNAs, which will match messenger RNAs that are complementary in sequence, and then target those RNAs for degradation or for silencing and prevention of translation. And this is an ancient, well-conserved pathway that was actually used to protect against viruses. And we can actually manipulate that in the laboratory to study gene function. We were initially injecting RNAs into mosquito embryos and looking at nerve cord phenotypes, doing comparisons of how some of your axon guidance genes would, if you knock them down, what happens, and getting some really neat nerve cord phenotypes. We could compare gene function in Drosophila to other mosquitoes, and it was very interesting. And you can see some of this work was coming out around 2011. What happened after that is we had some initial success with granting, and then we had a lot of cutbacks in scientific funding, and it became really hard to get funding. And so we had to start to get more creative. And I was very frustrated one day with funding and the translational direction it was taking, and I thought, well, okay, maybe I should just join this and see what happens. If we say, what if we use these small interfering RNAs that we're injecting to mosquitoes and really try to go about using them in the field as pesticides, hopefully in a manner that is species specific and where we can just kill mosquitoes. And that has been our guiding hypothesis. We got a bit of money from the Gates Foundation to start this work back in 2015. And what we did was large-scale screens. We were injecting embryos. We actually also figured out ways that you could just soak, well, actually it had been published by others, that you can soak the larvae in sRNAs and actually knock down genes. And we did these large-scale screens and identified hundreds of larvicides, so pesticides that kill larvae. And a subset of those we would find would have 21 or 25 base pair target sites that were conserved in multiple species of mosquitoes, Aedes, Anopheles, and Culex, but were not found in non-target organisms. If things hit a fruit fly, I would toss them out and we wouldn't push forward on it. We had a number then with some money from the DoD that we learned would kill both larvae and adult mosquitoes too. And so we were particularly excited in the subset that would kill multiple species, not non-target organisms, no matches there, and that would be genes required in larvae and adults. We then pushed on and looked at multiple delivery systems for these RNAs to the mosquitoes. We looked at chitosan nanoparticles, bacteria, and the one that's highlighted here is yeast, because we've really pushed forward on this system. We think it is the most promising. So I'll tell you a little bit more about that. Some of the advantages of it, as you know, you eat it all the time, and it's used in the food industry and the beverage industry. We know that you can actually express RNA in the yeast and heat inactivate it so that you're not putting out any live GMOs into the environment. And so I think that's critical that it would be classified as a dead microbial. We think it's going to be cost effective. We know that larvae, and actually it looks like adult mosquitoes too, really are drawn toward the yeast, so it's serving as a bait as well as a mechanism for producing the RNA because of the genetic tractability of this genetic model organism. It's used all over the world, so we thought that it would be something that could be adapted to resource-limited countries. It's a major billion-dollar worldwide industry, so the production should be scalable. There are many different formulations of yeast, liquid yeast, dried yeast, so those are good for insecticides. And we're really looking for ways that we can seamlessly integrate the use of yeast with ongoing mosquito control measures. So not much on the genetics here, but Saccharomyces cerevisiae is very genetically manipulable. We expressed short hairpin RNAs that were identical to those small interfering RNA target sequences we'd identified in our screen. Express that in yeast. We heat and activate the yeast. We can dry it and then feed it to larvae, so we can make it these little tablets out of the yeast, feed it to larvae. These were highly effective. We can just do larvicide assays, usually with 20 larvae is a nice little way of testing the larvicides. That's the WHO recommended protocol. And a 50 milligram tablet will kill 20 larvae. We've also discovered recently that we can actually feed the yeast to adult mosquitoes, and I'll talk a little bit about that today, but that was a really exciting discovery in the lab. So 3 directions. I'll talk a little bit more about our larvicide work. I then want to talk about the adult mosquito feedings, and then at the tail end I'll get into the mosquito sex separation studies, which are really becoming quite an interesting study in our lab. So just a little bit on the nomenclature. When you see our genes in this talk and in our papers, we would typically call them by the gene name. So here's our Aedes aegypti larvicide targeting a gene called FEZ2, or LRC is a different gene. These are some nervous system development genes. The dot and then the number is actually the number in our screen. So FEZ2.52 was larvicide number 52 targeting this gene FEZ2. We have larvicides that will target just Aedes aegypti. Some of the ones that would target multiple species of mosquitoes as a larvicide, here are 2 examples which we've published on. One that impacts, targets synaptotagmin and semaphorin. So a lot of nervous system development genes, that's my specialty, so we've tended to characterize things that function in the nervous system. So these were broad range larvicides. Then our final group, sort of our favorite group, are the ones that will act as both a larvicide and adulticide and target multiple species of mosquitoes. And we've really taken these hundreds of larvicides and focused in on about 5 and characterized those in detail as our top targets. One is Shaker 463, targets a Shaker channel, which is well characterized, of course, in Drosophila. And then Dope 1462, this was actually a gene that Dr. Hill's lab had characterized years ago. We have a larvicide and an adulticide targeting that, which we published on recently too. And the ones that we had identified and characterized in detail do not, at least in silico, have target sites outside of mosquitoes. That was one of our criteria. If we look at some of the yeast larvicide activity for these larvicides, you can see we get upwards of 90% in those trials. And the control I'll mention here, and it'll be true throughout the talk, this is a control yeast gene that expresses an interfering in our RNA molecule that has no known target site in mosquitoes. So it does express RNA, doesn't target anything. That is our control here and throughout the studies that I'll present today. So these look pretty good. We, of course, can go back into characterizing gene function. So this is the data from that Dope 1462 larvicide, targets a dopaminergic receptor. And this is just a marker, NC82, for active neural activity within the brain. You can see when we silence that gene in larvae, we lost a lot of neural activity. And so this is a marker we often use in our studies, just to show you that we can go on and characterize modes of action for our insecticides. And that's really a forte of our laboratory and a primary interest of mine. We can do outdoor assays. So these are very controlled environments where nothing gets in or out of our studies, that we can do on rooftops of buildings at Notre Dame. Just make sure if you take these yeast tablets and subject them to the environmental stresses that they'll still function. And as you can see, we still get very good activity and killing rates in these semi-field trials. When we were funded by the USAID, it was a Zika Grand Challenges project. We set up this diagram in our grant, and it's really been a guide for many of our studies in recent years. We wanted to take this work from the lab to the semi-field assays, and that's what I've shown you so far, and eventually expand to larger field trials. Trinidad is one of our primary field sites. Thailand is another. And as you'll see, we have engaged with consumers and stakeholders in Trinidad to understand better their acceptance. We've done a lot of refinements and optimization. We knew at the beginning, and that's why these arrows were drawn in both directions, that we were going to have to do a lot of refining to get this working. And so we realized that early on. We also knew safety was going to be critical, and I'll speak to that a little bit today, and eventually getting out to these large-scale field trials, hopefully with involvement of industry partners. And I do have a patent application in, and I mentioned not to make money off of that. That's my goal, not my goal, but to make sure that these mosquito control measures we're developing will get out to the people who need it worldwide, and that was our reason for patenting this. All right, so I would usually organize the talk at this point into some key questions that have come up during talks like this one, or just as we were developing these strategies on our path to commercialization. Could it be suitable for application through conventional pesticide application systems? Is it something that can be seamlessly integrated? Well, we sort of designed it that way from the beginning, looking at things like these mosquito dunks that you can buy on Amazon used to treat water storage containers. We picked the dried yeast and sort of aspired to have this same sorts of formulations, and I think that we designed it that way from the beginning, so hopefully it'll be true. Can you use the pesticides against field strains? We're using lab strains in the data I'd shown you previously. Are strains that are resistant to some of those commercial pesticides in the pictures I showed you in Puerto Rico, do they work? What about multiple species? So we can take field strains of mosquitoes. Here are some that were begun in Trinidad, and we saw that our larvicides worked really well here. These are actually resistant to a larvicide that's typically used there called Temaphis, and our yeast larvicides work just fine. And we've actually taken a lot of those larvicides I introduced at the beginning and published data across multiple species, including Aedes albopictus, Anopheles gambiae, and Culex quinquefasciatus, and shown that the larvicides work across multiple species of mosquitoes. What about different types of water? Tap water, what if something's growing in the water? What about different size containers? How many larvae can you have in a container? So we've spent a lot of time on this. We need to spend more time. But we've done a number of different containers, from these 1-liter containers up to 30-liter containers. We saw even in different densities of mosquitoes, hundreds of mosquitoes, or those 20 that we were using our initial assays. As long as you put in enough yeast and the larvae can swim to it, they will do just fine, and the larvicides will work. So these different size containers shown here have not been a problem to us. Those large water storage containers I showed you the pictures at the beginning of the talk, what if you go up to that size, so more like 200 liters? Here we may have reached our limits. What we found just in control assays is that those first instar larvae, the tiniest ones, don't tend to swim to the bottom of the containers and eat yeast that sinks down to the bottom. And so we've begun to address this by varying the buoyancy of our larvicides. And we have some really great collaborators at the Southwest Research Institute that are helping us address this problem. Some of the formulations for those mosquito dunks that I was showing you early on, those actually float. So we might be able to think about those sorts of floating formulations. And I'm not going to show you data here today, but I believe we've addressed this problem now and have good data and good formulations that'll address these larger storage containers. So we're encouraged by that. What about resistance? And this particularly came up because, as you saw, we're killing about 90% of our larvae. What about the other 10%? Are they resistant? So we did some work to address this, and we found actually, even if you select those 10% that live and let them breed, out to 11 generations we've gone, we don't see any changes in susceptibility to the larvicides. So at least not in our hands, we have not found resistance yet. We actually know that if we raise the larvae as individuals, we'll get 100% kills all the time. So what we believe is happening is that the larvae start to eat each other as they're dying rather than eating our yeast. And if you remove that possibility, then they only eat the yeast and they all die. So, and I think this is not abnormal for some larvicide assays, but we think that's the cause of that 10%. Whether that's going to play out that way in the field, we'll have to see. We're learning more. Several people have wondered if we've thought about expressing multiple different short hairpin RNAs targeting different genes in yeast. And I think we have done these sorts of assays to some extent in the lab. We don't see an increase to 100% kill rates when we do that. But I do think that this could be important in the field if we're trying to prevent resistance. So we've always thought thought we would get into that direction, and I think it'll be important as we try to commercialize this to use our yeast, which is of course genetically adaptable, to express multiple hairpins. Any impacts on non-targets? Well, I've shown you in silico that it looked good, but in practice, what happens? And so we've at least got some select arthropods that we fed our yeast to. We don't see any significant changes in their survival, and some of those assays with that dope 1462 are shown here. Of course, this isn't going to replace, going out and doing those toxicity assays that the EPA requires on vertebrate organisms. We're sort of waiting until we have a final commercial formulation to do those sorts of studies, because they're very costly, and you don't really want to do that work until you know what you're going at for your final commercial formulation. So we need to do that eventually, but we're not quite there yet. And we've just expanded our different types of organisms we use for our non-target assays, just some data across our top 5 favorite larvicides and adulticides here. Really not seeing any significant death to date. End user suitability, I think I have a whole 2 slides on this, but it's actually been a few papers that we've published in recent years where we've gone into communities, online surveys in Belize, and then more recently studies in Trinidad, where we engage with the consumers in community engagement events. We do paper surveys, we interview participants, and this has really been a rewarding part of our studies because we can really learn quite a bit, and use this as a way to educate people about mosquitoes. But big take-home messages here is that people are really concerned about safety and efficacy. The efficacy, of course, feeds into the price, and they're concerned about aesthetic things like, does it stink? Things gonna grow in my flower pots if I use your yeast? Those are all things that are important to them. We thought that the GMOs might be an issue, and we really are not finding, at least in our engagements in Trinidad, when we explain to people the yeast is heat killed when we use it, there aren't significant concerns. So we'll see if that pans out as we look into different areas of the world, but it's been encouraging so far. Really unanimous support for using our larvicides. People were asking us, okay, will you use my property as a trial site? That's the sort of engagement you want when you're developing a new control tool, because people, it's only going to be as effective as the buy-in. If people aren't going to use it and aren't going to use it properly on their properties, it's not going to work as a control intervention. So having people early on participating in the process has been key. We also got a lot of critical feedback from people, wanting to know how long it was going to last. Could you make formulations that would last for a month or 2 so I don't have to put new tablets out all the time? That would help keep the costs low. And so we got great recommendations. We took all of them into consideration, and they're letting them shape our project. So the people's feedback is really making changes to our project. In terms of the storage stability and shelf life, this was a big consideration early on. And take-home message is that we really could go in and develop these dried formulations with our partners at the Southwest Research Institute, looking at different ways of drying, to the point where if you would take the yeast through an accelerated storage, which is an EPA test, you heat the yeast up to about 53 Celsius for a couple of weeks. It's supposed to be equivalent to 6 months of storage at room temperature. We finally got formulations that worked well after being subjected to that treatment. We've gone in, we're making commercial packaging that should even improve that shelf life. And so we've done a lot of work in that direction.
Molly?
Yeah.
I'm sorry to interrupt.
Sure.
I really am. I'm thinking maybe your slides are not advancing. I don't know what other people are seeing, but I'm still, yeah, might just be frozen.
Okay.
Oh, okay.
Again.
Sorry.
All right, let's just start it from the current slide. I'll try sharing again and see if that solves it.
It was showing the non-targets, and I suspect.
Oh no. You lost me. Okay, what about now?
Yes. Good.
Okay, good. Let me know if it happens again. Sorry about
Will
that.
do.
Okay, so what about if we take that yeast and put it in water and store it? How long will it last in that water? And we've actually gone in and done multiple releases of larvae over 5 months now and shown that we still get decent killing activity. As long as there's enough yeast in that container for the larvae to eat, it will keep killing them for up to 5 months. So we were very encouraged by those results. We had money from the DoD too. Again, if you're thinking about using this as a control intervention, you better know a bit about oviposition of behavior of mosquitoes. If you treat the water with the larvicide that the mosquito is repelled by, it's not going to lay eggs there, so it's not going to do you much good. And actually, we were encouraged to find in our yeast-treated containers, so we put yeast in containers in Trinidad, did big studies with hundreds of ovitraps over a 6-month period with our collaborators there, and found that overall the yeast treated containers would gather more eggs than those that were treated with water alone. And so this was great. That means that maybe we can lure the mosquitoes to lay eggs in these buckets treated with yeast. We did find in the context of these assays that these long-lasting formulations that would last for months were going to be very critical for these ovitrap assays because we don't want to go in and have to add tablets to the yeast. And this was, also our stakeholder engagement was telling us those same things. That was part of the reason we went in and spent time on that new formulation development. Of course, as soon as we got the new formulations developed, Trinidad was in really bad shape and remains to be in bad shape during the pandemic. And we were talking, it's so hard on the grad students and all of us now with the pandemic. The grad students down there have been in lockdown for months and really haven't been able to get back to their projects for this whole time. So it's sometimes it's useful to hear these stories about people from everywhere, but of course, it's been hard on all of us, but we're hoping to get started down there. The vaccine rollout down there is occurring, and I think we're hopefully going to get started up there soon again. Is the production scalable? These are the things when you're training, am I going to need to learn how you ferment yeast at scale? Wouldn't have anticipated that. So a big advocate for interdisciplinary training. I had to learn about it. So I did what I could do by writing a review article to learn more about the process of fermentation. So I'll refer you to that paper if you're interested. What we found is that those lab strains we've been using really don't grow very well in pilot fermentation studies. You want to grow things on molasses, which is the cheapest carbon supply that you can do. And so if you're growing at scale, it's critical. Lab strains don't do very well on those. Here's your Red Star yeast that you buy at the grocery store. That's really your benchmark. So we know that we need to develop more commercialized yeast strains. Another thing we learned during the process, we were actually expressing things with an inducible promoter. As a geneticist, this is something you always do. Turns out the galactose inducible promoter we're using, galactose is super expensive. So people that ferment yeast for commercial purposes never would use that. So we need to go in and change our promoters, probably put in multiple hairpin RNA constructs under a constitutive promoter as we develop these tools. So a lot of efforts are being put toward making those commercial yeast strains now. Is the cost competitive? I just got a request for a project report from IVCC. How are you addressing this? We need to know what our final formulation is first, but we need to work with economists and people that are good at projecting what the cost of something is at scale. It's a critical thing, and I don't underplay it. I just don't have the tools to address this question myself, but you don't want to invest a lot of money into something that's not going to be cost competitive. So the cost of current larvicides is really sort of a benchmark that we have to keep in mind. Regulatory concerns, of course. Eventually, you're going to have to ask for a project registry from the EPA. And until you have that final formulation, you're really not at a point where you can do that. So we're on hold with that. But one thing that we can do is that you have to do your testing in U.S. strains of mosquitoes. And so we've gotten some eggs collected in Florida. We've been collecting them in Indiana and Michigan last summer and this summer. And here we are are collecting this recent weeks. We can test our larvicides there, and then eventually we're going to have to do some US field trials too. So this is something that's in the future. Okay, I'm going to switch gears a little bit, talk about our adult mosquitoes. And we really wanted to use the same approach of developing things in Aedes aegypti and applying it then to other mosquito species. And we received some money from the DoD to begin to look at things like sugar baits. And mosquitoes, when they're not drinking blood, the females drink your blood, of course, they are actually sugar feeding. And people have begun to capitalize on this. IVCC is working with a number of groups to try to put pesticides into sugar feeders that will then kill the mosquitoes. And I think the way that we can help this project is that our pesticides are more specific to mosquitoes, as I've shown you. So there's a lot of interest in this. We've developed little systems for feeding our siRNAs to the adult mosquitoes. We went through our hundreds of larvicides and fed them to the mosquitoes in this way, and identified initially 8 and then got down to those 5 broad-based mosquito pesticides that kill larvae and adults, which I had mentioned to you. We are looking at ways for delivering these, and then for this project, we're interacting with folks in Thailand. We are actually looking to expand this to Trinidad and eventually to Africa too. And we've gotten some more DoD funding approved to help with those studies. This is the movie link I gave you. So when we feed those small interfering RNAs to our mosquitoes with the sugar baits in the lab, we got some really cool phenotypes. The one in the movie in the chat box is when we targeted Shaker, so it's kind of fun to look at. You can see the kill rates we were getting, and then the time that it would take to kill mosquitoes. So it's not real fast acting. It does take a few days for the mosquitoes to die, but they do die. We're getting really nice kill rates, upwards of 90%. The really exciting development then was somebody said, hey, could you try feeding the yeast to the adults? And we were like, not thinking this will work, but sure, we'll try it. To our pleasure, it actually works quite nicely. You can put the yeast, just mix it in your sugar water, put in a red dye, which is actually quite attractive to Aedes aegypti. And this isn't blood. This is mosquito that's eaten our red dyed yeast. We're getting really nice kill rates here. The great thing is with the small interfering RNAs and sugar bait, we are getting about 50% of the mosquitoes drinking the sugar water in the laboratory. With the yeast, they love it, and they'll flock to it within a few hours. They all become engorged with the sugar meal and eat the yeast. 100% feeding rates that we're getting across multiple species. Great kill rates that we're seeing. We can do nice dose response curves as a good insecticide person would do. And so we're really characterizing this. Our first paper on this has been submitted recently, and there should hopefully be that one and a couple more in the near future. So we're really excited by these results. Just to show you, with some money from IVCC, we've been able to go in and repeat these same sorts of experiments in malaria vector mosquitoes. So here's Anopheles gambiae. It's feeding with our small interfering RNAs and the sugar baits. They too will drink the yeast-baited solutions and get great kill rates there. So we're very excited about this. Data from the paper that we submitted. This is targeting a gene called RB-Fox. Here's our Adulticide data, some of the Anopheles gambiae data, Culex, Aedes albopictus, all are really significantly killed by our yeast adulticides fed in the sugar bait. We did go in and look at the larvicides as well, and we get nice kill rates with our yeast as larvae as well. So looking good, and I think the money from DoD has been approved to continue some of that work. We were just starting to get the field work going in Thailand when the pandemic hit, so they at least were getting some of their assays to repeat our studies working in the laboratory before they were shutting down too. So we're hoping to really push forward on this with some DoD funding when things get better there. But a great group led by Along Palawat in Thailand at the AFRMS, the military base. Again, our U.S. collections of mosquitoes for eventual EPA approval are going to be critical here too. So we're doing those adulticide assays with our local mosquito collections as well. And just a reminder, this is our whole list of questions on the path to commercialization that I took you through for the larvicides. We're going to have to ask all those same questions once again with our adulticide assays. So we're pushing through those and checking those off and pushing forward as best we can, and eventually hoping to get these things back out into the field as well. Okay, in the last few minutes, I just want to take a little bit of time to talk about our sex separation project. So this is a project that my postdoc Lim Pyrai had really wanted me to work on. Could we use the system to produce male mosquitoes? So why would you want to do that? There's actually quite a bit of interest in this for a number of reasons. If you look at some of those Wolbachia trials that they're using, and then things like gene drive and some of these transgenic mosquito approaches, a large number of these population-based control strategies that are being tested now require the release of male mosquitoes. If you look at the transgenic trials they're doing in Florida right now, they're releasing males. People don't want to be bitten by females that are released in mass. Males aren't going to bite. So it's a lot more, if you look at your stakeholders, there's a lot more buy-in from people if they know that you're just getting male mosquitoes released. But to do that, if you're releasing thousands of mosquitoes, you need some way of getting rid of the females. And unless you want to sit there and sort them under a dissecting scope for hours upon hours, which actually people do that. If you want to make that easier, you got to figure out things and ways to do that. Now, Google, if you look, Verily has developed a nice camera separating system, which is great. If you try to set that up in other parts of the world, that might be challenging. So things that would work in a remote, resource limited region of the world is necessary. And so, of course, our yeast looked quite interesting from that standpoint. So long story short, we wanted to do screens with our sRNAs just as we had with the larvicides and looked for things that left with males and few females. If they looked good in a screen, we could go ahead and produce the yeast strain and then feed it to the larvae and end up with just males. But what genes? What Lim asked me this, and I was like, oh, we're doing all this field work with the USA grant, Lim, go away. And finally, I said, okay, I'll look at the M locus, the sex determination locus in Aedes aegypti. Had been resequenced the genome, and they got some really nice sequencing in this hard to sequence region of centromeric region of chromosome 1. And I said, okay, I'll think about it a little bit. And I was looking one day at this region in VectorBase and noticing all these long non-coding RNAs, these genes. What are these? I said, what is a long non-coding RNA? Okay, these are things that, of course, don't encode proteins. They're about 200 nucleotides or less. They have, if there's a process in a cell that has been described, they've been shown somewhere to regulate it. So we don't really know what these are doing in mosquitoes at all. They've at least been annotated in Aedes aegypti. That's not true across all the different mosquitoes or insects in general. And they certainly hadn't been functionally characterized. So this piqued my attention. I said, okay, Lim, we'll go ahead. We'll try to target a couple of these and see what we get. And lo and behold, we started targeting these long non-coding RNAs on chromosome 1. And we got incredibly distorted sex ratios. So here are some of the yeast larvicides we made. And as you can see, we're getting really nice female killing and very little impact on males. So I said, well, this is actually quite interesting. We got some NIH money to pursue this. Here are data just from a couple of our best larvicides that were recently published. What we wanted to do is confirm the silencing. So yes, we silenced the target genes. That looked good. Remember that if you're going to release these males, they need to be able to compete well with males in the environment for mating. This is true for the Wolbachia projects, for the transgenic projects too. So mating can't be impacted by the RNA treatments when the mosquitoes are in their larval stages. And we saw that the mating percentages looked good for our larvicide treated mosquitoes. And we saw that the females that they had mated with laid eggs. The fertility and fecundity looked comparable to control treated male matings. So this all looked quite good to us. Could we scale this for mass rearing? So in mass rearing facilities, larvae are reared at much higher densities and larger trays. And the good thing is that the larval diets that are often used in these rearing facilities actually include yeast. So we have that going for us. Dr. G at Michigan State has done a lot of work on Aedes aegypti and has a nice mass rearing diet. He gave us his protocol. We basically manipulated it to the point where we could put our yeast into the diet rather than his normal diet, which contained just regular dried yeast. And we could get nice female killing at scale too with 2 of our top larvicides. So we feel like we're on to something. We don't anticipate that this is a standalone technique. And maybe for Aedes aegypti, where you want, 0.001% females, and they've actually, got your Verily camera system that'll give you that, maybe it's not going to be your top thing for Aedes aegypti. Possibly, as you looked at different parts of the world, it could at least help in combination with some other strategies. But if you look at some of these other mosquitoes, Anopheles, different species of Anopheles, or Culex, where we don't really have good sex sorting strategies. The camera system is not being adapted well for those species. These are going to be things that are going to become more critical. This is just to show you, I didn't mention this yet, but at scale, the males were performing quite well. Looking at their wing sizes is a correlate for fertility and fecundity, which is typical in the field. Okay, so getting into this question, what about these other species? What could we do there? Unfortunately, the long non-coding RNAs are not conserved, or at least haven't been sequenced in other mosquitoes. So we don't have good orthologs for those, but we do have several protein encoding genes in the region of the M locus. One of these we published on recently, MTNB, which is part of the methionine salvage pathway. And here's some data from Aedes albopictus, Anopheles gambiae, Culex quinquefasciatus. When we target those genes in these mosquitoes, we actually got nice sex separation too, particularly in Culex and Aedes albopictus. Anopheles gambiae, it worked all right. We got about 60% female killing. We could probably do better than that. But it does seem to be a female-specific requirement across different mosquitoes, which is quite interesting because these mosquitoes have very different, reached different parts, gone to different aspects of chromosome, sex chromosomes. Anopheles gambiae has a true X chromosome. Culex and Aedes mosquitoes are believed to just have that sex determining region that on chromosome 1 that we were looking at a moment ago, and haven't really evolved full sex chromosomes yet. So it's interesting that this works. And I think there's some interesting implications for evolutionary biology that I'm happy to discuss with folks. We're trying to scale these things up right now. Aedes albopictus, we're getting large cultures like we did for the Aedes aegypti, still getting about 4-to-1 female ratios with no significant impact on wing size as a correlate for fertility and fecundity. Anopheles gambiae, some of our other hits actually are getting nice separation there, so about 5-to-1, so we have better hits than the one that I showed you in the previous slide. The mass rearing is actually in progress as I speak, so I can hopefully comment on that soon. Culex quinquefasciatus is also looking good. We've got about 5-to-1 ratios, a good male wing size. We've also developed separation for Culex pipiens now too. And in addition to the important things like West Nile, the Culex vector, we've been actually approached by a number of folks that are quite concerned about the bird populations in Pacific Islands. For example, Hawaii, where the crested honeycreeper and the scarlet honeycreeper have been dying due to mosquitoes that are vectoring pathogens that are killing these birds. And so we may have some interesting implications for the birds in the Pacific Islands. This is just, I want to give you some time for questions, but just checking our boxes. This is an article by Phoebe Pathanos talking about what you would want for an effective sex sorting strategy. And I think we, due to many of the good things about yeast systems, can check a lot of those boxes. Again, probably not going to be a self-contained system for sex separation, but one that could certainly be used in combination with things like, hand separation. I'd rather separate 5-to-1 male to female ratios than 1-to-1. So I think we could really help folks in that. We're doing much of the same sorts of project development plans as I discussed for the yeast larvicides in terms of the female-specific larvicide plan. Some of the future directions, what are the mechanisms here? I'd love to think about them. Is there dosage compensation at all that's evolving in Aedes aegypti? People would say not, but I think some of our data suggests actually that there might be. What are those non-coding RNAs? They have all these different functions that have been described in cells. What are they doing in Aedes aegypti? We don't know. What if we target the adult long non-coding RNAs? Could we get adult female-specific killing? And then, since you're an entomology department, we really need to annotate long non-coding RNAs in other insects and functionally characterize them. So a call out for the need to do that. We are just one thing. Everybody wants this magic bullet. Will RNAi be the magic bullet for killing mosquitoes? We can't stop doing source reduction. Other things, looking at surveillance of insecticide resistance, all of these things have to be done in an integrated mosquito management, keeping in mind that we want things to be culturally and biologically acceptable and least toxic. And so we're really keeping this mindset. I want to give you a chance for questions, but particularly wanted to point out Keisha Mysore, who's done most of the work that I showed you today. Lim Hapire, a former postdoc in the lab, was also key in starting particularly the Adulticide assays and pushing me on the sex separation. Really talented group of technicians. Particularly want to thank Dave Severson, a close collaborator, all of our colleagues in Trinidad and in Thailand, and formulation specialists, Southwest Research Institute with Craig Duckham. And thank you also to the funders, and happy to take your questions.
Thank you so much, Molly, for that really amazing presentation. It's just an incredible scope of work with so many amazing potential impacts and really fascinating basic science as well. So thank you. You're my hero. I was monitoring the chat, and I will provide that question to you. But before I do, just I encourage others to pop your questions in the chat. And I will put them to Dr. Doom and Scheel. And if not, then at the end we can raise hands and Eric can unmute us. So the question from Matthew Dittman, how important are the artificial water sources in creating, it's a great question, in creating larger populations of mosquitoes? And that's as opposed to natural water sources.
Yeah, so actually quite important. So this was addressed by Dave Chady, who is on our slide. He actually was our collaborator in Trinidad, longtime collaborator of Dave Severson, and died right at the start, right before the USAID grant was awarded. So that was unfortunate. His graduate students have continued with the project there. Nikela, Keela, and his technicians have continued to be our collaborators. But he had published some really nice papers in the 90s, and then looking at surveillance in the early 2000s, and looking for different containers, how productive they were for Aedes breeding sites. And these larger cisterns and then those larger water storage containers actually are pretty major contributors to the Aedes aegypti populations, at least in Trinidad. It's critical to do those studies throughout the world, though.
Okay, thank you. I've got a question along those lines, but I'm going to take the questions from the floor first. This is from Mike Scharf. He says, awesome talk. What do you think could be the mechanism of sex distortion for the non-coding RNA? Yeah, that's a tough question.
For the interest of time, actually, I may still have that slide in. I think I even tucked it in. No, I took it out. It's interesting. ROX1 and ROX2 in Drosophila are non-coding RNAs. They're probably the best non-coding RNAs that have been characterized in mosquitoes, and they're involved in dosage compensation in Drosophila. Exists also has a similar function as a non-coding RNA in humans for dosage compensation. I get in trouble if I mention dosage compensation in Aedes aegypti, but something is clearly going on. I actually think, and I'd love to get this, the vector control doesn't like to fund molecular mechanisms, unfortunately, but I'm really curious what they're doing. I wonder if there's some dosage compensation that is starting to evolve as those sex determining regions on the first chromosome in Aedes aegypti are evolving. If we look across the different species of mosquitoes, and this is a mechanism, a sex specific lethal is conserved, they must evolve pretty early as those chromosomes start to evolve towards sex chromosomes. So I think from an evolutionary standpoint, this is an interesting thing to think about. And I would love to hear more ideas on this because I'm incredibly interested in this from a basic science standpoint.
It's too bad. As soon as I hear you talk about this and chromosome location, I think immediately of someone who has retired from our department. That's Professor Jeff Stewart. But he would love to hear this, and
Absolutely, I
I
should
think it
talk to
would,
him.
yeah, it would be great for you. I'll connect you because I think he'd love to chat with you on this as well.
Good.
And spent a lifetime looking at Hessian fly, which is not that different from mosquitoes, right?
Right.
So another question from Matthew Dittman. This is a great question too. We're using sugar water for the adulticiding, cause problems by attracting other pests, such as wasps, etc., etc.
Right.
Yeah.
So if you look at Westham has done some work with IVCC, and they are really looking at this for targeting anopheles in Africa, and have done a lot of studies that look quite promising. What they have done is gone in and developed a sort of a membrane feeding system where only insects that can probe into it, like a mosquito, are capable of drinking the sugar feeding. They think that it will help with the pollinators in that fashion. But if we can bring something like our system into that, I think we could even further that. Ideally, we can have honeybees that would drink our RNA and not be impacted at all. And some have called for us to do some testing in honeybees. It's too bad because Dave Severson used to raise bees, and that was his background, was actually in bee ecology. And he retired recently, and I was trying to get him to get his bee boxes back out. I think we need our final formulation, though. Those tests on toxicity on the pollinators are quite pricey, so we'll stay tuned.
Yeah, thank you. I want to follow up with you on Westham. For those of you who don't know, it's a company, right, that's developing attractive, toxic, or targeted sugar baits for mosquito control with the Innovative Vector Control Consortium. I don't know what their active ingredient is for control, but I see they have some product in the product development pipeline. I'm curious about that and how
they've
well
published a bit with Gunter is his name. I can look up some of those publications too. Gunter Mueller.
Okay.
A few things have been published by them.
Okay. Well, I've got lots of questions, but we are coming up on the hour on our time. So I'm okay. I'm going to ask it very quickly, and
I can stick around.
I'm just curious your path to commercialization from here. Are you thinking about product development partnerships or a company taking this on? Are you thinking about spinning out yourself? I mean, do you? Seems like you'd never have you must be bored, Molly. What can you be? What can you do? Time to start a company. So
I have to say, during the pandemic, I did think about spinning out myself a couple of times. As you're waiting for contracts and things to get signed and these commercial partnerships to develop, it's a really slow process. And sometimes I'm like, oh, if I just do this myself, it would be faster. If you look at things like, what stops me, look at Roundup and those huge lawsuits and things like that. So, what's your commercial model, probably to make something that somebody else can produce that could have the amount of insurance required to cover that sort of thing. I didn't really want to start a business. A lot of the USAID grantees did start businesses. I didn't feel called in that direction. If I did it, it would be more of a means to an end. I mean, I'm really a basic scientist at heart, so to do this translational work is already a stretch for me. To open a company would be another, but it certainly continues to cross my mind for sure.
Well, thank you. And thank you for sharing the insights to philosophy and thinking and career path. It's great for our students and postdocs.
Sure. And as you mentioned, I was at a small liberal arts college for a while too. So if any of your students are thinking in that direction, I'm happy to discuss that at lunchtime if they want to pop in.
Okay. Well, thank you, Molly. Really amazing talk. And thank you so much for visiting with us today. As I said, there's a 2 to 2:30 open discussion session. If anybody would like to join that, just email me and I'll send you the Zoom link. But let's give Molly a virtual round of and we look forward to welcoming you back in person. We can really go and have coffee and a nice meal somewhere.
Thanks everyone for your attention. I look forward to that as well.
Okay, thank you. And thanks to Eric and everybody, and we'll see you later.